For centuries, reductionism has been the dominant scientific approach. Break a complex system into its smallest parts, understand each component, and reconstruct the whole. This method has been incredibly successful, driving advances in physics, chemistry, and even molecular biology. However, when applied to complex, far from equilibrium systems like the brain, ecosystems, or living cells, reductionism runs into a paradox. It fails to capture emergent behaviors, non-locality, and variability. Imagine trying to understand a hurricane by analyzing individual water molecules. The molecules follow physical laws, but the hurricane's swirling, self-organizing structure emerges from interactions at a scale larger than its parts. Similarly, biological systems, whether a neural network, a developing embryo, or an immune response, function not just through their components, but through dynamic feedback, adaptation, and self-organization. Consider variability, long treated in reductionist science as noise or measurement error. Classical science assumed that if we measured something precisely enough, whether body temperature, heart rate, or gene expression, variability would disappear. Yet, variability is often not an error, it is a feature of life. Your heart doesn't beat at a fixed rate but fluctuates slightly, responding dynamically to external conditions. Neurons in the brain don't fire identically every time but vary in response to subtle changes in experience and environment. Such fluctuations often enhance function rather than hinder it. For example, slight randomness in gene expression allows bacteria to survive antibiotic stress, and small variations in neural activity contribute to learning and creativity. Reductionist approaches also assume linearity, that small changes in input lead to small changes in output. However, in biological systems, small perturbations can trigger massive effects. Why does one genetic mutation cause catastrophic disease while others are silent? Why do some neural signals result in consciousness while others do not? These outcomes depend on complex networks where feedback loops amplify or dampen effects, often unpredictably. One striking example is developmental biology. Classical genetics assumed that if we knew the DNA sequence, the blueprint, we could predict how an organism would develop. But genes do not work in isolation. Their expression depends on dynamic interactions with their environment. The same DNA in different conditions can lead to entirely different forms, as seen in identical twins who develop different traits over time. Similarly, neuroscience once embraced the idea of grandmother neurons, the idea that a single neuron might be responsible for recognizing one specific face, like Jennifer Aniston. However, the brain does not store information in single units, but in distributed, overlapping networks where meaning emerges from collective activity. No single neuron knows Jennifer Aniston. Rather, patterns of activity across many neurons encode recognition flexibly. The failure of strict reductionism in biology has led to new paradigms: complexity science, chaos theory, and network biology, which recognize that systems evolve far from equilibrium, shaped by feedback, self-organization, and emergent patterns. Instead of asking, "What are the parts?" modern science asks, "How do interactions among parts create new properties?" This shift isn't just theoretical, it is reshaping medicine, engineering, and artificial intelligence. Can we predict how cancer cells evolve resistance, how brains rewire after injury, or how ecosystems stabilize after disruption? The answers lie not in studying isolated components, but in understanding dynamic, interdependent processes. Reductionism gave us powerful tools, but to truly understand life, we must move beyond it. The future of science lies not in simply dissecting systems into smaller pieces, but in uncovering the principles that allow them to adapt, evolve, and self-organize. The very properties that make life so astonishingly complex. Reductionism is the idea that understanding a system requires breaking it down into its smallest components, assuming that the behavior of the whole can be fully explained by studying individual parts in isolation. While this method has been successful in physics and chemistry, it faces limitations in dynamic biological systems, where interactions are complex and unpredictable. For example, molecular biology explains DNA sequences, but it fails to fully predict how an organism will develop due to complex gene interactions. A city's function cannot be understood by analyzing individual bricks in its buildings, just as understanding genes alone does not explain life. Reductionist models assume that small changes in inputs will result in proportional changes in outputs, yet biological systems are nonlinear, meaning that tiny variations can trigger massive, unpredictable effects. This makes it impossible to predict biological outcomes simply by knowing initial conditions. For instance, altering a single gene in an organism may have unexpected effects due to cascading interactions within genetic networks. A butterfly flapping its wings in one part of the world might lead to a hurricane elsewhere, just as small biological changes can cause large-scale effects. Reductionism assumes that what works at a small scale can be linearly scaled up to explain larger systems. However, emergent properties arise in biological systems, meaning large-scale behavior cannot be deduced from studying small parts alone, making predictions unreliable when moving between different levels of organization. Cellular metabolism, for example, follows different rules at the single-cell level than in multicellular organisms, making simple scaling ineffective. Studying the behavior of a single ant does not predict how an entire ant colony will organize and function. Reductionism suggests that biological systems follow a deterministic set of instructions that define their mature state, yet biological development is shaped by environmental influences, random mutations, and self-organization, leading to significant variation. This makes it impossible to determine a fixed outcome from an initial state. Identical twins, for example, share the same DNA, but can develop different physical traits and health conditions due to environmental factors. Building a house requires a strict blueprint, but growing a tree depends on unpredictable factors like sunlight, soil, and wind. Reductionism treats variability as unwanted noise that should be eliminated for clearer measurements, yet in biology, variability is essential for adaptation, resilience, and diversity, ensuring survival in changing environments. This means that variation is not a flaw, but a crucial feature of biological systems. Immune system variability, for instance, allows populations to survive diseases by ensuring some individuals have resistance to new pathogens. A jazz musician's improvisation creates new and unique music, just as biological variation drives evolution and adaptability. Biological systems do not require central control to form ordered structures, as they self-organize through local interactions. This emergent behavior means that the whole system exhibits properties that cannot be predicted by studying individual components. Unlike machines, living systems constantly adapt and evolve without a predefined design. Flocks of birds, for example, coordinate their movements without a leader by following simple interaction rules with nearby individuals. A traffic jam emerges from driver behaviors without any single person controlling the entire road system. Biological systems rely on feedback loops where outputs influence future inputs, making regulation dynamic and context-dependent. These loops create stability but can also amplify changes, leading to sudden shifts in behavior. This non-linearity prevents reductionist models from accurately predicting long-term biological responses. The human body, for example, regulates blood sugar through feedback between insulin and glucagon, and disruptions in this loop cause metabolic disorders. A thermostat adjusts temperature based on feedback, but if the system is too sensitive, it may oscillate between extreme hot and cold settings. Early neuroscience research suggested that the brain processes sensory input in a strictly hierarchical, linear fashion, yet later studies showed that the brain's structure includes feedback, cross-connections, and distributed processing, contradicting simple reductionist predictions. This reveals that perception arises from complex, dynamic network interactions rather than sequential data processing. The discovery of Jennifer Aniston neurons in monkeys initially suggested highly specific recognition, but broader studies found that perception depends on collective activity across neural circuits. Understanding an entire book requires more than analyzing individual letters, just as neural networks encode meaning through distributed patterns. Modern scientific approaches recognize that living systems function as dynamic, interconnected networks rather than simple sum-of-bots mechanisms. Systems biology integrates computational models, big data, and nonlinear interactions to study emergent behaviors. Unlike reductionism, this approach acknowledges that biological phenomena depend on relationships between components rather than isolated units. Cancer research, for example, now considers the tumor microenvironment, metabolic interactions, and immune responses rather than focusing solely on individual genetic mutations. Understanding a rainforest requires studying interactions between climate, soil, species, and ecological cycles, not just individual trees.